Hello, my name is Dr. Roderick L. Rowe, and today I will be talking about B cells. We are still on acquired immunity, and this is the third line of defense. Each B cell, or you can also call it B lymphocyte, generates a single type of B cell receptor, or BCR. What does that receptor look like? It has six polypeptides. If you look at the picture, 1 and 4 is called the light chain. 2 and 3 is called the heavy chain. And 5 and 6 is embedded in the membrane. The variable region that binds to the epitope is part of 1, part of 2, part of 3, and part of 4. This is the B cell receptor. B lymphocytes have multiple copies of this B cell receptor. Each B cell receptor only recognizes one epitope. B cell receptor has two variable regions. I mentioned before, 1, 2, 3, and 4. On the very tips of those polypeptides, you have a region that is changeable. They call that the variable region. That is the portion that binds to the antigens. The entire repertoire of an individual's B cell receptors are capable of recognizing millions of different epitopes. How did you make those millions of different combinations? Through this alternative splicing. Endocytosis occurs by the antigen presenting cell and processes it. If you forgot that, go back and review the presentation on pushing P. During pushing P, the first P was preparation, making the receptor. The second P was processing. So the antigen presenting cell devours the foreign entity. After it devours it, it will then process it, chop it up into fragments. Then it will present it. When it presents it, it will present it on MAC2 molecule. I drew a picture of what that looks like. My one tooth Pac-Man is called an antigen presenting cell. Let's say it is a dendrite. It will devour that bacteria. It will uh, process that bacteria, and then it will present. Those are your push and P, three Ps. It presents it on MAC2. The little purple circle is an epitope, a particle of the original bacteria. The epitope slash MAC complex then interacts with a T helper cell. So the APC has a catcher's mitt with its prize. It's baseball. That baseball is the epitope. T helper cell has a glove as well. Its glove is located on a CD4 molecule. They're going to bind together and hybridize and create a immunological synapses. In the picture, I have a red arrow showing you that synapses. CD4 of T helper is binding with MAC2 of APC. I drew a picture of this. CD4 of T helper is going to interact with the MHC2 of the APC. In this case, the epitope is transferred. The baseball is given to the T helper cell. It's transferred. I tried to illustrate that. I took the purple ball from MAC2 and gave it to CD4 on the T helper. Now the T helper differentiates. It transforms into T helper 2. Now the T helper 2 has the baseball in its glove, in its CD4 glove. And now it is going to interact with another cell called the B cell. It's going to create another immunological synapses. I drew a picture to illustrate that. The baseball was on CD4 of the T helper 2. It will now create a synapses with the B cell, MHC2. Now you see the B cell has the epitope. Remember, some white blood cells, like the B cell, has MHC2. CD4 of the T helper cell, T helper 2, binds with MHC2 of the B cell, creating that synapses. That's presentation of push and P. Look at the picture. I have a red arrow showing you this synapses. Notice there's another cluster of determination of differentiation molecule there. So the particle, epitope, was given to the B cell. A synapsis occurred there. And remember, it was another CD there. It was another CD there that created a better hybridization. Clonal selection happens when a naive B cell encounters that antigen. So when the CD4 binds with MAC2, it's going to activate the B cell. The B cell was a virgin. It had never been touched. Its glove had never touched a ball. The T helper 2 brought the ball on the CD4 and bound. When it's bound, the B cell to that T cell, T helper, a stronger immunological synapsis is created because of the CD40 receptor. This second signal activates the B cell to rapidly start to divide through a process called mitosis. This clonal expansion of the B cell only occurs after activation by the antigen. That glove had to obtain a ball. It had to obtain an antigen or an epitope in order that had to create the hybridization in order for it to be activated. Now that it is activated, it produces a bunch of different types of B cells. Here's one example. You're going to produce some memory cells. So some clones will form memory cells, which may give an enhanced response to subsequent exposures. Some clones will turn into regulatory B cells. These will minimize an immune response by secreting interleukin 10. Interleukin 10 is able to suppress the T helper cell. If you recall, the T helper cell, T helper 2, is the one that started all this by binding to the B cell, giving it the epitope, starting the B cell to produce the memory, the plasma, and the regulatory. All you need to do is silence the T helper 2. Interleukin 10 is released from the regulatory. It goes and tells T helper 2 to stop. And then now you've suppressed that signal. Most of the clones do not form memory and regulatory. Most of the clones form plasma cells. And these plasma cells are the ones that release the antibodies into the bloodstream. Those antibodies are the receptors. So these secreted receptors are called immunoglobulins. The immunoglobulin is different from the B cell receptor. In the picture, I put them side by side so you can see the difference. The immunoglobulin has 4 polypeptides, whereas the one that is bound to the membrane has 6 polypeptides. The immunoglobulin is also called an antibody, and the antibody that is made from that B plasma cell is only for the baseball that originally touched the glove. So that original antigen that bound to that naive B cell, every antibody it produces will target that antigen. And those antibodies are plentiful and they have different types. Antibody M, A, G, E, and D. So you can call it MAGED. Those are 5 different types. We will discuss these 5 types shortly. What are the functions of the antibodies? You've always heard that they kill. How do they kill? Or how do they immobilize? How do they neutralize? How do they stop the infection? Number one, they activate complement. Previously, when we talked about the classical pathway, and I've attached a picture here, I said that in order to start the classical pathway, you bind C1. C1 has to bind to the bacteria. What it first binds to is the antibody. So the antibody binds to the bacteria first, and then this complement 1 binds to the antibody, and then C1 activates C4, turning it into C4b and C2a. So we went over this previously in the unit talking about proteins that are involved in the secondary response. So in order to get this activated, you have to have immunoglobulin M. That antibody has to be already made, already floating in the bloodstream, and it has to bind to the microbe. Once it's activated, C1 complement will trigger inflammation. Neutralization is when antibodies bind to toxins, rendering them no longer harmful. Found a cool picture to show you the toxin is the little triangle, the orange triangle. It wants to penetrate the yellow cell. But if an antibody binds to the orange triangle, the triangles cannot get to the orange cell. So technically, they have neutralized the toxin. So that is the second thing antibodies will do. Antibodies can also block adhesion molecules on viruses and bacteria, inhibiting them from binding to target cells. In this picture, we're looking at HIV, and it has these orange little wheels. And those wheels need to land on the road. So let's think of it as an airplane. In order for it to land on the road, it has to have the wheels. Notice how the antibodies bind to the wheels. Now HIV is not able to land on the surface, which would be your cell membrane. So this is how antibodies block adhesion of viruses and bacteria. The third thing that antibodies will do is they'll bind to antigens through opsonization, making them more recognizable. They will coat and adhere to the target cell. Opsonization causes phagocytosis to be more effective since the microbe is coated with specific antibodies called opsonins. So basically, the phagocyte is looking for something to destroy, but if you coat this epitope with antibodies, it's more likely to see it. It knows where to go. It knows where to adhere. It makes it more likely to adhere. So this next slide shows you that. Immunoglobulin G has a stem. So think of a Y. The stem of it is going to bind to the neutrophil or the macrophage, whereas the Y is going to bind to the microbe and immobilize it. So now you see that the antibody has a binding site on the neutrophil and a macrophage. 4, antibodies immobilize through agglutination. This occurs when they aggregate and cross-link a particle together. In lab, you did an experiment where you looked at fake blood. And when the fake blood agglutinated, you said, oh, that is blood type A or blood type B. What happens is cross-linking occurred and made particles fall out of solution to make it visible. So that is cross-linking and agglutination. The fifth and final thing antibodies will do is they will bind to the target and bind to a natural killer cell. So antibody-dependent cell-mediated cytotoxicity occurs when the stem of the antibody binds to the natural killer cell. So now the stem can bind to a natural killer cell, and some antibodies can bind to the stem of a neutrophil. But in this case, ADCC, this target cell is going to die via apoptosis. How about the structure of the antibody? Here is the antibody, and I've told you before it's made of 4 polypeptides, pink and green. The Y-shaped antibody is composed of 2 pairs of identical polypeptide chains held together by disulfide bonds. 2 pairs. One pair is pink and one pair is green. The symmetrical Y-shape arrangement forms 2 major regions, the FC region and the Fab region. The FC region is where the stem is, and the Fab region is where it binds to the antigen. Antigen binding occurs at the Fab region. In this picture, we're looking at electron micrograph of plant pollen. This pollen can bind to the variable region, the portion that changes. That is in the Fab region. The Fab region has a heavy chain and a light chain. The heavy chain is pink. Dark pink is the portion that is variable. And the green light chain, the dark green is the portion that's variable. The FC region is the stem. It's the anchor. It anchors to the membrane of phagocytes. Which phagocytes will it bind? Down in the picture, I have labeled macrophage, neutrophil, eosinophil. Those are the phagocytes that the FC region will bind to. So the FC region serves as the anchor for the phagocytes. The amino acid content does not vary in the constant region. So light green for the light chain doesn't change. Light pink for the heavy chain doesn't change. A pair of identical heavy chains are bonded together by disulfide bonds. So if you look at the heavy chains, they're in dark brown here. You see two disulfide bonds holding them together. Each identical light chain is bonded to one heavy chain. So I'm circling a light chain. It has one disulfide bond holding it to a heavy chain. And that's only on one side. The mirror image, the exact same thing occurs. There's 5 classes of antibodies based on the FC region. So immunoglobulin M, immunoglobulin A, immunoglobulin G, immunoglobulin E, and immunoglobulin D. The only difference is that FC region. So let's look at the antibody types. Here are the types: MAGED, G, M, A, D, and E. The very earliest secretion of the breast is called the colostrum. It is thin, yellow milk, and it contains high levels of immunoglobulin A. Colostrum is the most important meal of your life. This should be mandated by all parents. The first 2 days of life, that infant should drink colostrum. Why? Because it has high levels of immunoglobulin A. This means every time mom was exposed to an antigen, her B cell made antibodies against that antigen. Immunoglobulin A is for that antigen. Then she was exposed to another antigen. She made another immunoglobulin A. Then she was exposed to the third antigen. So a pregnant mother that just gave birth, when she breastfeeds, she's going to give that baby a whole bunch of immunoglobulin A molecules for all different types of antigens she was exposed to. The immunoglobulin A is going to coat the baby's GI tract and guard against infection from E. coli, salmonella, poliovirus, rotavirus. Later, we're going to talk about Streptococcus agalacea and how it causes neonatal meningitis. This can help reduce that, breastfeeding the baby. Immunoglobulin A is secreted in mucus and serous fluid. So it'll be found in saliva, intestines, nasal cavity, and so on. Vitamin A deficiency decreases immunoglobulin A response. So if you're a vitamin A deficient, your immunoglobulin A isn't going to do what it needs to do. Another important thing about vitamin A is that it is going to help enhance class switching. Instead of immunoglobulin M being present, you'll switch and turn it into immunoglobulin A when needed if you have appropriate vitamin A. Immunoglobulin G is able to cross the placenta. Vitamin A again, it's very important. If you're deficient in vitamin A, you decrease the response of immunoglobulin G. This was observed in mice. Immunoglobulin M has many binding sites. 1, 2, 3, 4, 5 in this picture. I have a side-by-side comparison of G and M. IgM acts as a B cell receptor. IgE is present with allergies or with parasitic worms. So if you have those, you have elevated immunoglobulin E. IgD or Ig immunoglobulin D also acts as a B cell receptor. In this picture, we have the vitamins that we need daily. We have our water soluble vitamins, and we have our fat soluble vitamins. I've extensively talked about vitamin A and how important it is in reference to the immune response. But you also have to be aware of the toxic levels of those fat soluble vitamins. Vitamin A, you're going to need about 900 micrograms daily. Pregnant women might need a little bit more, around 1,300. There is a such thing as too much. So toxic levels around 3,000 micrograms for adults.